And Cyrille is a research
Hello.
Is a research director at CNRS and a team leader at the Paris-Saclay Institute of Neuroscience in France. Cyrille obtained his PhD in neuroscience in 1998 at the University Louis Pasteur in Strasbourg. And he did his postdoc at the University of Maryland in Baltimore, and then came back to France, where he was recruited as a permanent scientist by the CNRS. So Cyrille's research projects mostly aim at understanding the cellular and molecular mechanisms of cognitive dysfunctions, intellectual disabilities, and neuropsychiatric disturbances in several X-linked genetic disease, including Duchenne muscular dystrophy. And this is how we started a happy and fruitful collaboration a few years ago. So today, Cyrille is going to talk about the rescue of Dp71 expression in the retina and the restoration of retinal function in Dp71 null mice after AAV treatment. So Cyrille, the screen is yours. Thank you.
Okay. Let me know if you can hear me and see the screen.
That's perfect. I can hear you and see everything.
Okay, thank you very much for inviting me to present some data in this symposium and for organizing this symposium. Thank you very much as well to Mirella for this very good introduction on the complexity of the dystrophin gene, which will allow me to go faster in my introductory slides as well. So today I will present you data showing first that the loss of one glial form of dystrophin called Dp71 alters Ergs, and in a second step that we have been able to have an efficient rescue of these deficits by using an AAV vector to derive ectopic re-expression of Dp71. Okay, so this is a work that we have published last year in IOVS, and this was actually in collaboration with Mirella Barboni, with Jan Kremers, and with the group of Alvaro Rondon, who indeed initiated this small network with a focused interest on the role of dystrophin in the nervous system, and that pushed me to be more interested in retina because my original interest is the brain, but there are many similarities and it's very interesting to compare retina and brain in this field. So I will go quite fast because Mirella already explained the complexity of the gene, and that depending on the position of the mutation, you can have cumulative loss of several dystrophins. And regarding the brain Dp71, it's important to note, for example, that the IQ of the patients is more and more affected when you have a cumulative loss of several dystrophins, and that the most severe cases of intellectual disability are in patients for whom mutations affect the expression of this short product of the dystrophin, which is short, but which is the main product expressed in the nervous system. And you have a comparable genotype-phenotype relationship in the retina, as Mirella perfectly explained just before. And similar observations have been made in DMD patients and DMD mouse models. The main phenotype being originally characterized as a reduced B wave amplitude, particularly in scotopic conditions. But again, for the retina, the phenotype is more severe when the mutation affects the expression of all dystrophin, including the Dp71. But one question was remaining, is whether a selective loss of Dp71 may in itself alter ERGs, and this was the purpose of the study I will present you today. So to address this question, first, I will just summarize again what you have seen just before with Mirella. Most of the dystrophins are expressed in all layers of the retina, particularly Dp260 at the level of the photoreceptor on bipolar synapse. But Dp71 is selectively expressed in the radial Muller glial cells and can be detected across all the retinal layers, even though it is enriched in the end fit of this Muller glial cell at the inner limiting membrane, but also at the level of collaterals contacting the vascularization. And at this vascular glial interface, it is known that Dp71 interacts with many signaling proteins and transmembrane proteins, including very important ion channels, the aquaporine AQP4 water channels involved in water homeostasis, and the Ki4.1 potassium channels, which also play a role in the buffering of extracellular potassium. And in this mice, it's been shown that the loss of Dp71 induces an increase in the blood retinal barrier permeability associated with inflammation, some capillary degeneration progressively, and also retinal edema, altered water homeostasis, and delayed potassium buffering. And all this has been studied mostly by the group of Alvaro Rondon at the Institut de la Vision in France, and you have some references here of this work. And the fact that the concentration, the extracellular concentration of important ions regulating the physiology of neuronal cells in the retina led us to think that the sole loss of Dp71 should have an important impact on ERG, on the retinal physiology. Okay, so to address this, we used the Dp71 null mouse, which has been originally created by the group of David Yaffe in Israel. This mouse has a selective loss of Dp71 because its promoter has been replaced by the beta-GO gene. Our objectives were several-fold. We wanted to know whether the loss of Dp71 can indeed impact ERG, which would mean that there's a role for Muller glial cells or maybe to the blood retinal barrier in the generation of normal ERG responses. And we also had the goal to see whether the deficits in these mice could be rescued by a gene therapy approach. So this is what I'm going to present now. To measure full-field binocular ERGs in mice, we have used the Gansfeld-Bold retiport system from Roland Consult. The mice have been dark adapted overnight for scotopic examinations. They were chemically anesthetized, the pupils fully dilated, and we used contact lens electrodes. We had 22 transgenic Dp71 null mice and 12 wild-type littermate mice, and twice the number of eyes because the recordings were binocular. As explained by Jan Kremers this morning, we used an extensive repertoire of stimuli and ERG parameters in order to have an idea of the cellular pathways that are affected in this mouse. So this included the recordings of scotopic A and B waves, photopic B waves, which involve the bipolar cells and photoreceptors, but also oscillatory potentials, which have been extracted from these flash ERG responses, and which may involve other retinal cells, for example, amacrine cells, and also sine wave and sawtooth responses, which can give us an idea of the integrity of the post-receptor cone system. So here you can see the flash responses recorded in a wild-type mouse. We did recordings at several light intensity, at 5 different light intensities, and we used a filter to extract as well the oscillatory potential from these recordings. And as you can see in Dp71 null mice, which are in dark in these different plots and curves, you can see that we found a reduced amplitude of the B wave in these mice, but no change in the amplitude of the A wave. And this has been found for all the intensities, as summarized here on this on this plot. However, the implicit times of these two waves were not affected for the A wave and B wave. There's no change in the implicit times, and there was no difference in the oscillatory potentials. So from this, we concluded that the on-bipolar cells may be specifically affected by the absence of Dp71 in this mouse model. Okay. Then we also recorded flash ERGs in photopic conditions. So you have an example of the recording in a wild-type mouse here. And as you can see, we also found a reduction in the amplitude of the B wave in Dp71 null mice in these conditions without any change in the implicit times. So this, our conclusion from this was that there's an altered cone-driven on-bipolar cell response. Then we addressed sawtooth responses in two different conditions, and you can see already in wild-type mice that the sawtooth responses are different between each others depending on the condition, if we compare mesopic and photopic conditions, but also they are different depending on the on or off mediated response that we record. Here you see the results compared between wild-type mice and mutant mice. And this was quite interesting that we had no change at all in the mesopic conditions for this ERG parameter. And we only found a difference in the mutant mice for the on-mediated responses in the photopic conditions. As you can see here, we had a significant reduction of the amplitude of the positive part of this recording. So we conclude that we have an asymmetric defect here involving the cone on bipolar cell pathway. Finally, we also recorded the sine wave flicker responses at different temporal frequencies from low to middle and high temporal frequencies. And we also found here a reduced amplitude of the response, but this was restricted to the low temporal frequencies between 3 and 12 hertz without change in the phase. And from this, still, I mean, on the basis of other studies, especially from the experience that have Jan Kremers with this kind of parameters, we conclude that we have post-restoral defects involving the cone system and involving sustained rather than transient retinal pathways. Now, the second step of our work was to determine whether it's possible to rescue, to compensate these deficits by rescuing an ectopic expression of Dp71 using an AAV vector. And we have used an AAV vector developed by Denis Dalcara at the Institut de la Vision, which was produced from directed evolution strategies in order to select vectors that could reach the retina from vitreous injections. So in this vector, we included the full Dp71 coding sequence, so without the introns, and an ubiquitous CBA promoter. However, the serotype was selected, was a CHH10 serotype, which has been previously shown to specifically target Müller glial cells in the retina. And we also added a 2A peptide to allow bicistronic expression of Dp71 on a GFP marker. So injections have been done in the vitreous, in the right eyes, and the left eyes were controlled non-injected eyes. We also injected some mice with a control vector only expressing GFP. Okay, so this is the previous results that have been obtained by the group of Alvaro Rondon with this vector in Dp71 null mice. As you can see, the GFP marker shows that the AAV vector specifically targets the radial Müller glial cells of the retina. And this allows a re-expression of Dp71, but also relocalization, of the Dp71 in the ELM and around the blood vessels in the different layers of the retina, and also a relocalization of the associated channels, aquaporin 4 and Q4.1, that are located where they should normally be located. This group also showed that the blood retinal barrier permeability, which is abnormally increased in Dp71 null mice compared to the wild-type mice, is restored to normal permeability after treatment with this Dp71 AAV. In our own experiments, we confirmed that, in fact, Dp71 is not just re-expressed with this vector, but is overexpressed as compared to wild-type levels, which we confirmed both at the RNA level and at the protein level. Then we performed the first control experiment, first to see whether GFP or the surgery itself could alter the ERGs. Indeed, one thing is that we have done this experiment 3 months after the initial characterization of the phenotype in the same animals. But you can see that 3 months after, we still have good ERG recordings, and that the presence of GFP because of the AAV vector, the control AAV vector, does not modify the ERG responses in wild type or in Dp71 null mice for the flash response as well as for the sawtooth or sine wave flicker responses. So there's, at least we can say that there is no light activated enhancement of ERG response that could contribute or bias the rescuing effect that I will show you just after that. Here you can see the comparison of the control groups. So we have, for the controls, we have pulled the non-injected eyes on those injected with control vectors because we did not find any difference between these different groups. And you can see that 3 months after this surgery and injection of the vector, we can still see the genotype differences, meaning this reduced B wave amplitude in Dp71 null mice, non-injected or injected with control vectors, compared to wild-type mice, but that after treatment of the retina with a Dp71 AAV vector, we have a full recovery of the amplitude of the B wave in these mice. And this happened for all the intensities that we have tested in this protocol. We also found a full recovery of the wave in photopic conditions. As you can see, after treatment, the amplitudes are comparable to wild type. So we conclude from this that we have a full recovery of the on-bipolar cell-dependent B wave amplitudes after treatment with this AAV vector. Then we also found a full recovery of the other parameters that were shown to be affected in Dp71 null mice. So you can see that we have no more difference in the sawtooth flicker responses or in the sine wave flicker frequencies after treatment. So we have a full recovery of the cone on bipolar system after treatment. So to conclude, we show that we have that Dp71 dependent mechanisms in Muller glial cells may likely contribute to the generation of the ERGB wave and other parameters by modulating the on-bipolar cone pathway. And we also show that ectopic re-expression of Dp71 with AAV-based strategy based on the transfer of the Dp71 coding sequence induces a full recovery of retinal physiological functions. So this may give the perspective to use this type of tool for gene therapy for related disease with blood retinal barrier breakdown or retinal edema, of course for DMD, but also for other disease in which Dp71 has been shown to be downregulated, and for example, with diabetic retinopathy. So, of course, I thank all the people that have been involved in this work, particularly the group of Alvaro Rondon and Denise Dalcara at the Institut de la Vision, Michel Roux at the IGBMC at Ilkirch-Grafenstaden in France, and the three groups that have been involved in this ERG study, the group of Jan Kremers in Germany, Mirella Barboni in Hungary, and the group of Dr. Ventura at University of Sao Paulo. And of course, the fundings. Thank you very much for your attention, and I'm ready for questions.
Thank you very much, Cyrille. Thank you for being on time, and thank you to all the speakers for being on time. This is really nice. So we have time for a few questions. I don't know if there are any in the chat. People can also raise their hands. You can stop sharing. Yep. I can maybe start, Cyrille, with one. So you showed that you had an overexpression of the Dp71, and that leads to a full recovery. Have you, by any chance, tried or happened to have tested lower doses of AAV that would have led to partial restoration of the Dp71? And if so, what was the phenotype? And or if not, what would you expect?
Well, that's a good question. In fact, in the previous paper by the group of Alvaro Rondon, when they showed the relocalization of the channels and restoration of the BRB permeability, they have used a lower titration of the AAV, but they also had an overexpression. So we should have decreased a lot more the concentration to avoid having this overexpression. So I don't know what would happen if, but if we had a lower concentration, but at least we see that there's no adverse effect. We have mainly a restoration of the phenotype. So this is at least a yes. Good point.
From a therapeutic point of view, I mean, of course, because we're working in the dystrophin field, and that dystrophin at the muscle level, we don't need to restore that much dystrophin. And we're actually, as you know, wondering about the level that would be needed in the brain. So I was wondering, yeah, in the retina, if we had any indication that only partial restoration could lead to functional outcome.
But we haven't tried this.
Okay. Any other questions for Cyrille? So the AAV that you were using, so it was the serotype, what was it?
SHH10. Yeah.
And that's specific to target the glial cells, so the Müller cells. Yeah. And if we wanted to target other cells in the retina, it would be different serotypes.
Yeah, I guess so. And this is, I mean, this is one of the objectives of Denise Dalcara at the Institut de la Vision, to select specific vectors, to select them to be efficient after injection in the vitreous to reach the retina, but also to reach specific cell types in the retina. So this vector specifically target Müller glial cells. From the nice images that they have made in the previous paper, you can see that the other stellate astrocytes are not targeted by the vector. I think it's a bit different if you use this vector in other tissues. For example, in the brain, it's not it's not so specific as what has been demonstrated in the retina.
Okay. Well, thank you very much.